Cancer. This word probably means something to you. Either you know somebody who's been diagnosed with cancer or you've heard about it on the news. There's many different types of cancers, but one thing they all have in common is that they are caused by uncontrolled cell growth. That means somebody's own cells grow out of control. Now, that doesn't mean that there aren't other contributing factors. For example, for some cancers, having genetic links, meaning it runs in the family, can make somebody more likely to develop it. Or having excessive exposure to UV light for some cancers. Those are risk factors that can increase your risk, but the major cause of cancer is uncontrolled cell growth. That also means it's something that is not contagious, and it's something that anyone can develop. You have to realize that all the cells in your body are very, very busy, and they're either in one of two different phases. They are either in a phase called interphase, and during interphase, they are just growing, they're replicating their DNA, they're doing their cell functions, or they are in mitosis. Mitosis is a fancy word that means that they are dividing, they are making new cells. Well, if you think about the cell cycle, whether they're in interphase or mitosis, it's kind of like a pie graph. They're spending about 90% of their time in interphase. Most of the time, cells are not dividing. Most of the time, cells are in interphase. They're growing, they're doing their daily cell functions, and they're replicating their DNA. Most of the time, cells are not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells, they do mitosis frequently, which is why your hair can grow up to half an inch every month. Mitosis is very important in order to generate new cells, which can help with growth, and also if you damage your cells. If you're walking down the street and you have some kind of accident and you skin your knee, well, you're going to need to repair those damaged cells. You're going to need to be able to do mitosis to make those new cells. The thing is, you don't want your cells to always be doing mitosis. Because if they're always doing mitosis, then they're always dividing, and then they're not carrying out their actual function. And that's a problem. That's where checkpoints come in handy. See, along the cell cycle, there are checkpoints to check that the cell is growing correctly and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. And what happens if the cell fails its checkpoint? The cell does something called apoptosis, which basically means the cell destroys itself. It self-destructs. This might seem kind of harsh, but actually it's really important because it ensures that you don't have these cells that are messed up continue on and divide. You don't want damaged cells to continue on and divide because they might not do what they're supposed to. They might start to grow out of control. They might not be regulated correctly. That's where cancer cells come in. See, cancer cells, as we had said, are your body's own cells that are growing out of control. So they're not following the rules of these checkpoints. They're flying past the checkpoints, going through the cell cycle, and frequently doing mitosis, which means that they're dividing very, very often. And so by doing that, they can grow into these things called tumors, which are really a mass of cancer cells. So we're not really sure what causes them to escape these checkpoints. There's a lot of risk factors that a person can have that can increase their risk of this happening, but when these cells get past these checkpoints and grow out of control, they become a problem because many times they can travel through the bloodstream, they start growing out of control, the body can't identify them and destroy them with the immune system. So this becomes an issue. And usually one of the treatments that somebody who has cancer will undergo is chemotherapy. And this works by targeting cells that go through the cell cycle quickly and hit mitosis quickly. And that's one reason why somebody often will lose their hair when they're on chemotherapy, because they're on this medication that's targeting fast-growing cells, and your hair follicle cells are also fast-growing cells. Cancer cells have some other factors that make them difficult to treat. One is that cancers have the ability to secrete their own growth hormone. This is very bad because growth hormone, in a sense, think of it as a hormone that says, feed me. And what happens is the blood vessels will divert over to those cancer cells and supply the cancer cells with nutrients. This is not good because this means the cancer cells will have the nutrients they need to keep growing out of control. It also can take nutrients away from nearby healthy cells. There's some medications that are being researched that maybe could stop the growth hormone from cancer cells. And in a sense, that would be great because the cancer cells would not be able to get the nutrients to grow out of control anymore. Of course, you have to make sure that the medication is targeting just the cancer cells and that you're not affecting the growth hormone of nearby healthy cells. You wouldn't want that. So it's kind of a tricky.
Is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So, ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So, 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase, M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into two cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. So interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23. Which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have 2 newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with 2 new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the 2 cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes and the ending cells only had 23. So they are not identical to the original and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study.